Thank you. So it took 170 years for the Royal Navy, when it was clear that vitamin C would save lives in people that were doing long distance travelling, to actually implement those changes. Nowadays, it takes 17 years for patients where there is a clear guideline for 50% of patients in America to be able to access the care on that guideline. We've got 17 weeks to introduce these genomic medicine centres, and hopefully in 17 minutes I'll tell you a little bit about it and what we're going to do. This just feels like a very psychotherapeutic talk. I've been waiting for this moment probably for 2 or 3 years, so thank you very much for the opportunity. Clinical genetics really is when mummy and daddy love each other very much and close the door, and it's about the genes that we inherit. But obviously it's incredibly exciting that we are also starting to look at somatic genetics at the same time. And clinical genetics obviously has been in services throughout the country. But I think it's wrong to say there's been no mainstreaming and that genetics is completely separate from the rest of the hospital. I mean, there can be a disconnect between the acute medical services, and that was alluded to earlier, and what we do on a Friday night in A&E, Monday morning in general practice, and the clinical genetic services. But look at the most common and deadly of all genetic diseases, rhesus disease of the newborn. We don't think of that as genetic. Same as high cholesterol. The two most common and most dangerous conditions, we just get on with it and do it. Now, how we think about consent, how we think about testing and mainstreaming obviously is going to change over time. And I'm going to talk a little bit about that, and also about where I see the direction of travel really when it comes to commissioning. So this is what we're going to talk about. How to find a genetic test, the rise of genomics, variance analysis, and really what I think of in the future about the commissioning. And where I really want to end up at the end of this talk is talking about genomic responsive records. It's one thing to be able to carry out the test. It's another thing to be able to interpret the test, and the bioinformatics I think is an enormous challenge that we face. But even when you've done that, to actually go from a result of hundreds, thousands, even millions of genomic variants to actually that A&E on a Friday night is still going to be a big challenge. And I think responsive records might be the way forward. Try and keep things simple, and we'll be talking about that. So where do people go to for genetics at the moment? Well, it's throughout that life cycle. It's all those things which you think are likely to be Mendelian, caused by a single change in a single gene. But obviously, our concept of what is truly Mendelian is changing. There's still a fundamental educational issue here. How many patients do I need to see who are going to die of breast cancer when they've gone to their GP before and they've said, don't worry, it's on your dad's side? How many people have to die when we still don't understand the basics of inheritance patterns? So we're talking about complex data. There are fundamental problems with the identification of inherited disease and the interpretation of genetic disease. It's really great to hear from people like Alex today about how we're going to try and work together and integrate, consolidate these services. But we do need to win that battle, I think, still in primary care and in secondary care, about the relevance of clinical genetics and somatic genetics. So I think it's work in progress. I don't think we've won all the battles yet. So the ladies in the audience are far too young to remember about 1980, but some of the men here might remember Ted Rogers and Dusty Bin, and the concept, I'll try not to abuse you here, 3, 2, 1. So this is a concept which we based on Lynch syndrome testing, but to try and give people a simple, universal idea of what an inherited trait is. And what we came up with is if you've got 3 relatives across 2 generations with one diagnosed under 50 with any type of condition, then you really need to start to think about whether or not this could be an inherited trait. GPs really quite like this sort of simple framework in which we can start to build an idea of trying to triage and identify patients more effectively. And we looked at 500 families, newly referred families, and found particular reasons why people came when they did. What we found is that a common death in the family was a trigger for health behaviour. A recent diagnosis is in the family, again, a trigger. But what we found is that 2/3 of the time, based on that 3-2-1 rule, an intervention could have occurred earlier. we found in 111 of the families, all the affected relatives had died. We were getting referrals with 5, 6, 7 people affected with breast cancer or ovarian cancer before someone had twigged, could this be genetic? This pathway now, in terms of the genomic sensors, is a huge step forward to try and get to people at that point of delivery. So fantastic step forward. So what do we do now? Well, we have a few, when we see somebody with a condition that we think could be inherited genetic Mendelian, well, we might start with Google. And one of my favourite places to start is OMIM, so Online Mendelian Inheritance in Man. And you put in your condition, and then you might get something like this, which tells you which genes might be involved and what are the criteria involved. Then from there, you might look at something a little bit more complicated, such as warfarin sensitivity. And again, a whole series of genes may come up, which gives you a clue. And this is where, for instance, a GP or somebody who's worried, could this patient's condition being genetic or inherited, might look it up. The problem with medicine is not the knowledge. It's easy to look this up. It's knowing when to look it up. Okay, with my students, we do a quiz. I give them a case of von Hippel-Lindau syndrome. And I always say, it's easy, it's the rule of 3. It's got 3 letters on the third chromosome, it's got 3 exons, 3% don't have a mutation, affects 3 parts of the body, 3 parts of the central nervous system. But you've got to know to look it up. When they look up retinal angioma and cerebellar ataxia, it takes them seconds to get the diagnosis. The art of medicine is knowing when to look it up. And that's how our whole relationship with knowledge is changing. So that's great, but people have got to do it. Okay, where can you find out more about the condition? Gene Reviews is very good. And you get all sorts of information here, which is very good, I think, for clinicians, even for some patients. And these are really good websites for people to use if they're thinking about genetic testing. UKGTN was the title I was originally given for this talk. And I said, well, there's going to be changes with the test directory. And they said, great, talk about that as well. So UKGTN, fantastic website where it has lots of information about the tests which are available and where, but also, as I mentioned in one of the questions, there's a gene dossier which explains why that test is available and the evidence for this. And so one of the pieces of information I'd like to take back to Genomics England is it would be really great to do something similar with the test directory, which I have to say, having looked at it, I actually think is very good. I think there's lots of very good and very sensible suggestions on there. But it's very difficult to rigorously and robustly analyse that data and without the evidence being published for it. So I think that would be really helpful. What does the UK-GTN do? Works within Department of Health and Commissioning support units and specialist commissioners and all of these networks up here. This is what it says that it does, UK-GTN reports and the laboratory membership, and it's a very good way of finding where a test is available. And then if you want to look up a lab, it's got all the lab details. And also if you look up a condition like Marfan syndrome here, it will then tell you about where it's available and all the different genes which are on that available panel. If you scroll down from there, it will then tell you more about the condition. So if people know about it, this is a very good system on UK-GTN. The difficulty, and I suppose why we are where we are now, is because this whole concept of single gene testing is going. And that's because the cost of a whole genome is going down, but obviously we have all these variants that are being described, and so the complexity goes up. And the idea of just picking off single genes is going. So ovarian cancer, and even, for instance, pheochromocytoma. Classically, I was taught in medical school, it's the rule of 10. 10% bilateral, 10% extraadrenal, 10% malignant, 10% familial. Well, actually now in our clinic, 60% of patients who are referred are Mendelian, have got a mutation. It's our biggest pickup rate. And as you were saying, endocrine is a really important aspect, cardiac, neuro, where there's a huge expansion in clinical genetics. But all sorts of things we're finding have got inherited components. The key, taking the family history, but also developing these pathology pipelines. Also, the use of non-invasive genetic testing, free circulating DNA, use of SNP technology for microbiology, for testing in pregnancy, non-invasive, also for looking for the way in which a tumour is changing as well, tumour evolution. So there's a huge change going on. But don't forget there's other type of chemical tests. So here's the cancer dog. The idea of a dog being able to smell urine and picking up kidney cancer. And what was interesting about this study is that one of the negative controls, the dog always kept on woofing at one of the negative control samples. And they went back to the patient, this control, and said, the dog thinks you've got cancer. And unfortunately, the patient did. But breathomics and metabolomics are going to be very important as well. And I suspect, I was asked recently, what is the NHS going to be doing in 70 years? And I was thinking, wow, what are we going to be doing in genomics? But of course, the answer is you'll be able to breathe onto your mobile phone. Or a little tab will come out. You put a urine or a blood spot. And it will be able to tell you your chromium levels for where your peak fitness, but also whether or not you've got cancer and whether or not your body is in perfect balance. So I'll be in trouble. So this was a slide that came out of the discussion event for genomic medicine centres 2 weeks ago for the accelerator site. I found this very interesting. The idea of the difference between simple, complicated and complex. And I certainly, bringing up a child, my daughter's 16 today, and I've tried simple and complicated, and the formulas don't seem to be working particularly well, but it certainly is complex. And I think it's important to say that developing a genomic medicine centre and service is complex. And the reason why it's complex is not only have you got to get the science right and the bioinformatics right and the sample pathways, you've actually got an emotional and educational barrier in primary care, in secondary care, because we've not won the battle. We need early wins. We need to actually be able to give the evidence. The reason why you need to give this drug and to do the pharmacogenetics is because this is the evidence of why. And once you've done that, you need to enable people to do it, and then I'm afraid you need a stick as well, okay? And these are the two sticks you're going to need. You cannot operate unless you have the right pathology. You cannot prescribe unless you've done that pharmacogenetic test. It's understanding, but with expectation. We have to enable, but it has to be both sides. And I think that's going to be the winning argument. We have to show the evidence, say why patients want and need those tests, and then we need to be able to enable it to happen. But the commissioners, and when I sit in front of my executive strategy board, they go to say, well, do we need to do this? The thing that will really make them listen is if they can't operate. Okay? And that's going to be the critical step to be able to make this a reality. Does anybody recognise this? Two things, amazing things happened in May 1997. One is some very unwise person allowed me to practice medicine. And the other thing was this. And I was coming on the train here today and I saw NHS computer sort of doom and sentence people to death. That was on the front of the metro this morning. And this is all to do with artificial intelligence and computers. Now, obviously, we generate huge amounts of clinical phenotypic data that we need to interpret, but also we need all those big genomic pipelines. And someone's got to bring them together. Does anyone recognise this for any particular chess Jedis? Yes? This is when man lost to the machine for the first time. So this is Game 6, Deep Blue, against Garry Kasparov. Garry Kasparov played, for those of you who are into this sort of thing, played pawn here, thinking that there's no way a computer would take this and smash open his defence. The thing about it is not that the computer was going to do it. The computer was programmed by a human to do it. Now, if you really want to really crack on and work with computers, you've got to do three things. The first thing is you only get out what you put in. So when doctors work with patients with artificial intelligence, one of the reasons why GPs are so good is that they listen to patients what they do say and understand also what they don't say. You have to look at your inputs. The second thing is that you actually need to sense check what is going on. We say, oh great, computers are fabulous, they're going to save us loads of time. Well, then people go off and do other things. You actually need to sense check that you're actually giving people mammograms when they need to have their mammograms. Somebody needs to be looking at that. Then you've also got to work creatively with the computers to develop better processes and keep that innovation cycle going. And on all three of those levels, yesterday you saw how it can go horribly wrong. We need to work with computers to understand the genomic traits, but it won't work unless actually we're together and actually constantly actually testing, are we improving what we already have, and actually publishing that evidence. So we talked about whole genome sequencing, and this has obviously gone into the idea of actually doing this because we need to get better access across the board, I think is a winning argument. There's no doubt that the majority of people who are eligible for genetic testing in primary care, and I think GPs have been missed out of a lot of these loops, most of the patients out there that are eligible for genetic testing don't get it. Okay, and that's an identification issue. It's because there's a perception they have other medical health needs. It's a perception that the doctor doesn't understand from an educational, or doesn't get emotionally the importance and the value of these types of genetic testing, as well as variations in pathology and bioinformatics. So I think developing a better system clearly is a winning argument. And there's been huge variation in quality, and Ian mentioned that earlier. And obviously, the development of the GMCs is a huge step forward, and I particularly would like to say that the patient involvement here has been excellent around issues of consent and result delivery, but also hugely powerful in developing these outreach models and work that we've done, for instance, with the Parliamentary Select Committee reports when it comes to how we're going to deliver genomic medicine and work with things like the Clinical Research Network in the future. So we talked about keeping things simple. So one of the things that we did was we developed the genome eligibility wheels so that, of course, there's hundreds of pages of criteria, but we thought the budding cardiologist will only want to look at 1 or 2 pages. So we came up with visual clues that could help people, they could stick on their wall in clinic, which would help them work out which patients are eligible. And that's for rare disease, but we also did that for cancer as well. And there's over 50 of those which are being used. There are a number of issues around data. Now, data is the new currency. It was interesting you talked about China earlier. We had recent discussions with the Chinese biobank. They said they would do 100,000 genomes for free. Okay, so data is power, and data is the new currency. Okay, and that's clearly how that data is stored and used is going to be important. As genomics gets cheaper, quicker, more predictive, and important for common traits, insurance companies are going to wake up. At the moment, we talk about Mendelian disease affecting 1 in 17 people with rare disease. Most of the time with clinical data, insurance companies at the moment can cover that cost. When we start adding in much more predictive power across the whole population when it comes for screening, for examples, for instance, in time there will come a tipping point where insurance companies will say this field is unfair. And that is understandable. Now, for small companies in America, you say this is a long way away. For small companies in America, this is already an issue for health insurance and going through Senate. So yes, we can have moratoriums, but there will come a time when actually people will not, insurance companies will struggle with that. But where is the data going to be stored from October? How will it link into genomic databases that Ian described? How will it link into GSIPs in the future? If something's found, how will that be then linked back into the patient? How much data will the clinician need to actually provide? Will the patient actually need to consent for additional findings at that point of delivery for the whole genome sequence? Now, those parameters are not, to me at least, clear from October. So some further clarification on that is going to be incredibly important. These are some of the additional findings which are being suggested at the moment. We've not received any of these additional finding reports, and that's been a frustration. And obviously these test directories are being brought in, but I have yet to see enough results to actually give, my impression is that they're very good, but I can't robustly measure patients' response against these additional findings because I've not received one, and we've recruited over 1,100 genomes. So these are the guidelines that are being developed around the variance interpretation. And I think there's a real disconnect between people's understanding of what is a DNA sequence and what is a clinical report. And actually that bioinformatic challenge. And it reminds me of the TV show, The IT Crowd, where the managers have got literally no idea of what is going on in the computers. And my concern is that the commissioners, it feels a little bit like, to me, to you, is that the commissioners, I think, talk about sequencing and they say, oh yes, we've sequenced all these genomes, but don't actually understand there's a huge difference between that and actually what are you going to do with your patient, what does it mean, what does it mean within that clinical framework that that patient has and all the ethnic variants. So classically in the past, we talked about how we interpret variance in 5 ways, looking at literature searches, segregation analysis, looking at the amino acid change, functional work and evolution. But now, and of course, we're working with our colleagues and our GMCs about, and nationally about how we will interpret these variants. And that has been a real bit of progress there, so that how sure are we that that variance's significance is being published? Variant tiering is going on in Genomics England. This is good. My concern is there's not been enough support financially for people to actually deliver this. The laboratories are being recommissioned at the same time as people doing their standard work and trying to add this work in as well. And at £200 per sample for patient identification, sample processing, IT transformation, pathology transformation, consent, for moving the samples around, for the bioinformatics, for the result interpretation, and to deliver those results for patients, taking into consideration the ethical and data requirements. £200 I think is a bit underpowered, to be honest, and I think to do this at the same time as this tiering is a lot to ask of the pathology departments. So I think that is one of the reasons why it's been slower than we would have liked. Things are improving, and this project is a huge catalyst for change in the right area. What we've done is we've done a lot of things at the same time. We're trying to do gene mapping, we're trying to do databasing, pathology transformation, working with patients and delivering a completely different way of thinking about clinical genetics and cancer, so that we will move to a world beyond microscopes, which is a fantastic concept, and we treat people for what the disease is, not what it looks like, with the right drug at the right dose first time. So that's all great, but you think about these big systems is that you only need one area of it to fall down and you run into a problem, and my concern, as said, is the bioinformatics. So these, in terms of the domains, with regards to genetic testing, obviously we're finding these solid gene panels, for instance. So this is what we think all of our patients will be able to access for solid tumours. And then there was a very good pipeline in to help to understand how that will then lead to an implementation and change in treatment pathway. This is coming out of my East of England GMC, with a number of patients with a number of variants which are interpreted. And it does take time. It takes time to review all of these variants, tiered variants. And this is hopefully where we will lead to, where patients, you can see here, with ovarian cancer, the vast majority of patients are given the opportunity to take part in a trial, and obviously somatic testing of ovarian cancer, very important for BRCA testing, for instance, because of the PARP inhibitor story. And what I would have done if I'd had my time again is linking with the clinical research network from the beginning, start with one rare disease and one tumour group, and show that in a rapid cycle the patient benefit, publish the patient benefit, learn from it, and then move on to another area. And what we've done is we've taken 400 pages of criteria all in one go, which is fantastic in terms of equity, has proved a challenge. In terms of the interpretation, huge news pipelines with databases, which will help us to have some transparency as to how we interpret our results. I think that's very positive. We've also got better databases, and here's another dog. This is what we do when we find a variant in BRCA1, BRCA2, or any of the cancer genes, and look at the published data behind it. So that's very much a step forward. And again, this is the new framework we use to be able to determine if that variance is significant. Academic networks are linked behind this. As the data is being published, I think this will become increasingly powerful. But I think for the first couple of years, I think the GSIP struggled to really see where their role was before some of that data became available. This is what Genomics England are saying that they've been able to do, and I think, as I said, there's huge progress that's been made, and it's been a great success. And it's certainly work in progress as we move towards the GLHs. And we've gone through all of that. I'm not showing that slide again. And what I just wanted to finish on really is thinking a little bit about drugs. We went to go and see this company recently who said that they want to be able to look at SNPs in about 20,000 patients or clients or residents in care homes, because their care homes are very interested in concerns about polypharmacy. And they were going to do this, as I said, in a private residential capacity. And I was interested around issues of consent for that, and also they were going to do this on saliva samples. A lot of people in care homes may struggle to produce those samples. But this is coming, and this company produces a 400-page book. Now, if a patient brings that 400-page book to their 8-minute appointment with their GP and drops that book on the desk, I wonder what the GP is going to make of that exactly. Obviously, in the cancer genetics group, we've been working at trying to develop these gene panels, but we need clarity on. So this was my psychotherapeutic moment where I dumped where I thought, and so I'll give you just a second to read that. So we've been asked by NHS England and Genomics England to tell them how we think this should be run, but we do need some understanding of the parameters, and I think that this is definitely still work in progress. So if you're asking for next steps, I think we need some of this clarity to be able to actually start to develop that roadmap. To develop the genomic network, responsive notes, what we've developed is a simple triage system based on the 3-2-1 scheme, and that a patient will go to a GP and say they're worried about their family history. GP will use a 90-second triage tool which will tell them if they're high risk or moderate risk or a population risk. Patient then gets a key to be able to develop and draw up their pedigree at home. That will then be sent to the genetics department. The system red flags potential syndromes. So if there's all the B's, breast, bronchus, brain, blood, and bone tumours, then it will say this could be Lee-Fraumeni syndrome, and it can automate letters. And we've got a sandpit where patient can access resources, all the information letters. We've got standard letters as well, and we want to use this for dynamic consent. So dynamic consent, patients can ask to receive feedback on research, access to new studies. I think dynamic consent is definitely the way forward for the CRN. In the East Midlands, we recruit 50,000 patients a year. The patients most likely to be wanting to be recruited to a future study are those that have already done so. So we could easily develop a system with dynamic consent for patients to say, yes, we want to receive additional information, and these are the topics we'd be happy to receive information on that might be relevant to us or our family. We used to run a medical supermarket model where we'd have dietitians, physios, smoking cessation teams, psychology, psychotherapeutics, research, the research network, clinicians. Patients would come there and we'd give them a series of talks, and then we'd have stalls and people could come and access what they're interested in. How many additional referrals did it generate? None. Patients go, they access what they want, and they say what they find out what they need, or say no thank you. It's as cheap as chips to deliver medicine in this way. We need to ask patients what they want. We need to put the patient at the centre of the commissioning model. We keep working in departments. Patient disease doesn't work in departments. We need to commission around the patient, and there are simple, cheap ways of doing it. So what we want to be able to move to is a way in which when your patient is sensitive to warfarin, you don't get a 400-page book landing on the GP's desk. Actually, it's flagged on the computer when the clinician prescribes the warfarin. That's the key. We need the notes to tell the patient when they need to be screened, because not purely on age, but on risk. And again, when we're looking at that complex data, the notes have got to respond to that data. So this is the step forward. This is the work of our colleagues in Finland were very interested in, artificial intelligence and developing genomic responsive notes. I think this is the way forward. Ask patients what they want. Make sure you understand what clinicians are going to need to be able to deliver this. Communicate effectively, particularly around issues of consent and data. Work with universal templates for business modelling, and develop these genomic results. There's a number of people here who have helped me over many years in my career, and I hope to also work with many of you in the future. I'd like to thank you for this wonderful opportunity to talk. I'd be happy to take any questions. Thank you very much.